Hi students, we are talking about the role of microbes in sewage treatment. And this part we were talking about that is a sewage treatment and the microbes. How far the sewage treatment just actually performed with the help of microbes. And at the same, we have to know the act enforced by the government to save water bodies. So how does the government normally just utilize certain acts to prevent water pollution and also save what is called the water bodies and maintain the quality of water? One such plan what we have, National River Conservation Plan. National River Conservation Plan, that is NRCP. That was enacted in 1995 by the government. What for? To improve the quality of water. To improve the quality of water. That is in the rivers, because rivers are the major resources of fresh water in our country. That's why now the government has taken the step to conserve such freshwater bodies. Now this is an assignment taken by what we have this NRCP. Now what are the actually just things normally done or to be done by what we have the NRCP? So the important assignment taken up under NRCP includes the following, just actually the programs. Number one, to capture the raw sewage flowing into the rivers through open drainage system, through open drains, and divert those sewages towards the treatment plant, for treatment. This is number one, capturing the sewage and then diverting the sewage to the treatment plant. Number two, for treatment of sewage, setting up a large number of sewage treatment plant for the diverted sewage to just break it into similar things. So, setting up sewage treatment plant for treating diverted sewage. So, a number of actually sewage treatment plants are to be just actually set up for treating, and they are doing that one, that is what we call NRCP. The third one, you have often noticed open defecation, open urination. To prevent such open defecation along the rivers, along the banks of the rivers, the government insisted, that is NRCP, to construct low-cost sanitation toilets. Low-cost sanitation toilets. What for? To prevent open defecation. That is elimination of waste on the river banks in order to prevent the pollution of water, to save the quality of water. So these are all the three steps actually taken up by this NRCP to improve the quality of water and prevent water pollution. Now the Ministry of actually that's Environment, that's a government. The Ministry of Environment, the Forest and Climatic Change. They have initiated what is called, this is Govt Action Plan and Yamuna Action Plan. What for? To maintain or to improve the quality of water in these two rivers and also in other rivers. To save the rivers, they have initiated what is called actually Govt Action Plan and River Action Plan. These are normally to clean the waters and improve the quality of water and save the rivers in India. Okay, let's talk about the Govt Action Plan and what we have Yamuna Action Plan, the next one. Now the Ministry of Environment, Forest and Climatic Change have initiated two plans to conserve water bodies. One, Govt Action Plan, another one, Yamuna Action Plan. What for? To save the major rivers in India. Now what is Govt Action Plan? It was launched on 14th January 14, that is 1986. What for? To improve the quality of water. How? Number one, interception, just breaking, not allowing the sewage water to enter. Number two, diversion, diverting what is called this polluted water, the sewage water, towards the treatment plant. And finally, treatment of sewage. After diverting towards the treatment plant, now the treatment of sewage, converting the sewage into a harmless material, either in the form of what is called pure water and then what is called the organic waste. And the second important just note for this one, the notion we can say about what is called the Govt Action Plan, to identify grossly polluting units. Grossly means major polluting units. What for? To prevent pollution in river waters, particularly in river Ganga. That's a major polluted river in India, you know that one. Even the dead bodies also released into that one as a sacrifice. And now that's why actually they are doing that process. The Ganga Action Plan was started to improve the quality of water in Ganga, to prevent sewage introduction, and also just diverting the sewage if any sewage is there, and then treating the sewage by means of treatment. That's what's called sewage treatment plan. That's about Ganga Action Plan. Now let's see what is Yamuna Action Plan. And that Yamuna Action Plan is what is called a bilateral project between the government of India and Japan. So two governments took part in that what is called Yamuna Action Plan. That's why I say it's a bilateral project. Now let's see Yamuna Action Plan. The second one, Yamuna Action Plan, it's a bilateral project between two governments, one Indian government, another one Japan government. So it was normally launched on April 1993, just the 1993 year. And what's the importance of this one? It was proposed to build up large number of sewage treatment plants. What for? Treating the sewage and discharge the treated water, the purified water into the water bodies like the rivers. That's the main purpose. So setting up large number of what's called sewage treatment plants so that they are being treated, that is the sewage materials are being treated and released as such, not as such, that is in the form of what's called purified water into the rivers. That's about Yamuna Action Plan. Study is under working and they are doing the process of cleaning the Yamuna River. Okay, that's about what we call act enforced by the government to save water bodies. Yamuna Action Plan, that we have what is called actually the Ganga Action Plan, NRCP and its role, etc. Now we have to go for the microbial fuel cell. Now next one, let's see about the microbial fuel cell or MFC. What is its role? So it is actually it is a bioelectrochemical system concerned with the driving energy with the help of bacteria or mimicking bacterial reaction normally found in nature. So mimicking bacterial interaction, you see that one, in nature we have a chemical reaction or interactions brought about by bacteria. The same one will be just mimicked. Mimic means being done just like that. Mimicking compound, you know that one. Mimicry, you know, that's the meaning. So it is now using mimicking bacterial interaction that normally found in nature. Now you see that one. What is the mechanism that is normally by which the MFC works? The MFC works by allowing the bacteria, normally see that one, to oxidize, that is, or reduce organic molecules. Oxidation reduction, you know that one, the redox reaction is called oxidation reduction. Now we studied in 11th standard the electron transport system where we have oxidation reduction occurs. So removal of electrons or addition of electrons through electron transport system. That's called as oxidation and reduction. Here MFC works normally by allowing bacteria to oxidize or reduce the organic molecules, either by means of what is called the normal respiration or by means of fermentation process. Now we have these microbes are formed associated with what we call the anode. I'll come this back. Okay, so here represented respiration, this is a normal respiration, the presence of oxygen. The products obtained are carbon dioxide, electrons and protons. Or by means of fermentation also the products obtained are carbon dioxide, electrons and protons. This is because of the oxidation reduction of the organic substances by the microbes. Two ways you know that one, either by anaerobic respiration or by means of aerobic respiration. In both the cases, we have the products carbon dioxide, electrons and protons. That is being brought about by what we have bacteria in it. So the bacterial respiration is basically one big redox reaction. That is nothing but oxidation reduction. That is, electrons are transferred. So oxidation occurs by the removal of electrons. Now the NAD, the respiratory enzyme, the electron carrier, being normally oxidized by the removal of electrons. You are getting what is called NADH+. while losing the electrons. Okay, now this NAD just actually while losing the electrons, normally you see that one, it becomes oxidized. So NAD, electrons are removed from the NAD, then it becomes oxidized, so that we have what's called NADH plus. When the protons are removed from the NADH, once again it becomes reduced. So either removal of electrons or what we have the addition of that is electrons, or removal of hydrogen or addition of hydrogen results in what we have reduction reaction or oxidation reduction reaction. That is called redox reaction. So in bacterial respiration, one big redox reaction, that is oxidation reduction occurs. That is NAD gets converted to NADH because of what is called oxidation, addition of oxygen, and it becomes actually reduced when electrons are added to NAD. So removal of electrons makes what is called this NAD to become oxidized. Once the electrons are added, it becomes NAD. So NADH plus is an oxidized form. NAD is a reduced form. So while fermentation is going on, or respiration is going on, we will receive the products carbon dioxide, then electrons and protons. Now what's happening? So in the case of bacterial respiration, it's basically one big redox reaction. There is both oxidation and reduction occurs, in which electrons are being moved around. Because of the reduction and oxidation reaction, we have the release of electrons and protons. Now the electrons are being moved around. Now what is happening? If you are taking the microbial cell, it's being separated into two chambers. One contains the anode, another one contains the cathode. And these two chambers are separated by a proton exchange membrane, simply called as PEM. Now what is happening? The microbe associated with what we have, microbe or bacteria associated with what's the anode, causes the reduction of organic molecules, leading to the formation of electrons and protons. Electrons and protons are removed. Now the electrons now pass through the anode to the outer circuit and generates a current. The electrons released here pass through the anode and resulting in the generation of current. At the same time, the protons are passing through what is called the PEM, proton exchange membrane, reaching what is called the cathode, where we have chemical or biological reduction of oxygen occurs. Chemical or biological reduction of oxygen occurs, leading to the formation of water. So here reduction occurs. So because of what we have the addition of just actually the protons, then oxygen, so it becomes reduced into water. And also we have the reduction may be either by means of chemical or biological reduction of oxygen, leading to the formation of water. So at the same time we see that when we are getting energy that is being used for generating current. That's why it's called as microbial fuel cell, as it is used for generating the energy. So in this chamber, we have the chemical and biological reduction of oxygen leading to the formation of water. Okay, now what is happening? You see, because actually the hydrogen atoms are added, that results in what's called the reduction reaction. So the anode associated with the microbes reduces the organic molecules into protons and electrons. Now the protons and electrons are released. This is what we have the microbe or the bacterium associated with the electrode. Inside which only we have the bacterial respiration resulting in oxidation reduction. Now the protons are passing through the PEM to reach the anode, to reach the cathode, causing the reduction of oxygen to form water. The reduction may be a chemical reduction or it may be a biological reduction. It's not necessary for it. They are using some reducing substance, ferrocyanate and ferric cyanate. These are all the substances. Here we have nitrogen, nitrates, sulfates, etc. being used in the case of biological reduction of what's called oxygen. That's not necessary. Okay, that's about that one. The electrons are passing through the anode to the outer circuit, resulting in the generation of electric current. And we have the protons are moving into the cathode chamber, and where we have there is a reduction of oxygen to form water. This is the principle used in the case of microbial fuel cell. So I will write down the result in just the remaining part of the microbial cell on the board now. So the microbes, namely the bacteria associated with that is anode, causes the reduction of organic matters or the molecules, that is by oxidation process. So it causes the reduction of organic molecules that enter into them and breaking them into protons, electrons and carbon dioxide, either through what is called the normal respiration, that is aerobic respiration, or through anaerobic respiration called fermentation. In both the cases, the products release are carbon dioxide, electrons and protons. Now the protons pass through the proton exchange membrane, reaching what is called the cathode, where it causes either chemical or biological reduction of oxygen to form water. Then what about the electrons? The electrons are passing through the anode to the external circuit and generate what is called current. Okay, the electrons pass from the anode to the outer circuit to generate the current. You see that one, the current being generated, that causes the lighting of the bulb. So that's about what we have the role of microbial fuel cell. And what is happening? The protons reach the cathode and causes the reduction of reduction of what is called, that is reduction of oxygen to form water. Here the electrons pass through the anode, reaching the outer circuit and generates electric current. That's the working mechanism of what we have, the microbial fuel cell. We are generating energy, hence called fuel cell, with the help of microbes by using what's called bioelectrochemical system. Okay, that's about that one. We'll talk about other things in the next class.